Hi everyone, I'm a new postdoc in the glass lab, and today I'll be talking to you about building living synthetic cells from non-living parts. But before I dive into the details, I want to take a moment to express my sincere gratitude to the Ruggles family, whose generous support is directly funding the project I'm leading. And without your contributions, this important work wouldn't be possible, so I'm truly honored to be a part of it. I also need to take a moment to acknowledge the amazing individuals whose hard work I'll be in part presenting today. These people have all done an incredible job in driving these initiatives forward, and it's a privilege to share their achievements with them. So without further ado, let's get into it. Beside it being one of the founding aspirations of the synthetic cell community, what's the purpose of creating a synthetic cell from non-living parts? On the foundational aspect of building our understanding, a synthetic cell would lead to new insights into how chemical systems organize and can be methodized to yield biological lifelike function. And on the practical uses, being able to boot up a synthetic cell with a full-size genome would allow us to facilely design systems capable of much more complex functions not found in nature for useful purposes in industry, agriculture, and medicine. So with the minimal cell, we have been able to boot up fully synthetic genomes, and others have created long-lasting protocells and even reconstituted phages from their genomes in cell-free systems, all of which suggest building cells from non-living parts should be possible. So our approach will be to use mycoplasma as a basis to construct the simplest organism that could grow in laboratory media. Our goal is to instruct the synthetic cell with a minimal cell genome and to provide the non-living energy sources and replication machinery from Mycoplasma capricolum in a transcription translation system, or TXTL for short. Finally, we will insert both of these into a synthetic lipid liposome designed to resemble capricolum membranes in lipid composition and size. So I'm going to break down how I'm going to accomplish this in the following sections. Developing mycoplasma cell-free expression, or CFE, showing the non-living parts fabrication of the liposome and a secretory system, and then the incorporation of components and genome installation. So making a mycoplasma CFE system proved to be exceptionally and unexpectedly difficult. For years, it was a failed effort trying everything ever used under the sun to make bacterial TXTL systems. And we ended up finding mycoplasmas have potent RNase activity on the surface of cells, which Zoomer mentioned, that rapidly degrades the cytoplasm and the ribosomal RNA in, the moment it comes into contact with the cell surface. So the team working on this project even went so far as to publish a negative results paper to cover all the things they tried so no one else would spend so much time trying to make a TXTL system for mycoplasmas. But as a last-ditch effort, our collaborators at the Radboud University, graduate student Andre Sake and his advisor Wilhelm Huck, attempted to try a technique from the 1960s using nitrogen cavitation in this cell disruption chamber called a nitrogen bomb. So by dissolving nitrogen into the cells and then instantly decompressing the cells, they explode, leaving liquid cytoplasmic extract and the cell membrane and all the RNase activity in a foam. So counterintuitively, this is actually a comparatively gentle method that only exposes each cell once to disruptive forces. So that actually avoids heat damage and oxidation. So if you're working on a finicky cell-free expression, this is a method I would recommend looking into. So this method proved effective in producing a TXCL from JCVI Syn3A. So centrifuging the lysate resulted in this foamy layer containing nearly all the RNase activity in the cell membranes, while the remaining lysate was usable for CFE. Andre Sake then optimized the system by adding additional exogenous mycoplasma tRNAs and utilizing an active machine learning method to iterate through and predict the best experimental compositions. However, we believe more improvements can yet be made. and this is part of what I plan to reproduce and work on. For instance, E. coli and Streptomyces venezuela systems can produce multiple levels of magnitude more protein. And I now have a nitrogen bomb of my own, and getting our system to that level will involve tweaking the growth, disruption, and the reaction conditions even more. So I have a lot more to talk about, but for more details on the CFE, I highly recommend going to the third minimal cell workshop and watching Andre's presentation. Next, I want to explain why we think installing the cell-free expression into a synthetic liposome will work. I will be able to tightly control the composition and stability of our synthetic membrane by adjusting the ratios of phosphorylated fatty acids and varying the cholesterol concentrations. And James Sayen's group, who spoke at our last session, has provided a great blueprint for the composition I'm going for by defining the lipidomes of the minimal cell and mycoides, showing that when grown on certain diets, the complexity of the final membrane can be reduced. So mimicking the membrane of M. capricolum might not be enough. So to provide the metabolic influx and efflux needed to sustain our non-living TXTL activity, it might be necessary to install a membrane protein translocation secretory system in the vesicles before the genome is added, so newly transplanted transporters can be inserted into the membrane. And in 2004, Vincent Nero demonstrated how one could solve this energy and material limitation problem and increase the capacity of a TXTL in a vesicle system by including the alpha hemolysin pore protein from Staph aureus to create selectively permeability for nutrients. And compared to a bulk reaction expression life of 2 hours and 5 hours after putting it in a vesicle that has some intrinsic permeability, adding this pore protein allowed for sustained and elevated expression for up to 4 days, with a protein production of 30 micromolar. So, to provide the metabolic influx and efflux in our system, I plan to install transporters in the lipid envelope by including a plasmid encoding transporter protein genes and a pre-protein translocase complex. So this method was recently developed by Willem Huck's group to install a E. coli sec-YEG secretory system. And I plan to either use this or tailor a plasmid for mycoplasma secretory complexes. So putting it all together, I can use microfluidics to extrude our lipids through an oil immersion and encapsulate the TXTL mixture with the secretory transporter plasmid. So I plan to start by creating mycoplasma-sized vesicles, but as the Pullman group spoke about last session, sizes of even 1 micrometer might be necessary to fully encapsulate all the machinery necessary for a Syn3A cell. Finally, once I have our non-living parts assembled and functioning, I can attempt to boot up the synthetic cell with a minimal genome. So there's 2 methods I plan to try. First is the genome transplantation, which has been well established, in which a gently isolated naked bacterial genome and the liposomes are mixed with polyethylene glycol and calcium to initiate the fusion. The other chemical transplantation method we have in mind is to fuse 2 lipids containing the SYN1 genome and the TXTL system. Our collaborator Kate Automala has recently developed a method called programmable liposome fusion to initiate this in an easily controllable way, which I'll explain in the next slide. So this works by anchoring complementary DNA molecules attached to their outer membranes. And this DNA duplex formation actually physically drags the outer leaflets of 2 liposomes together to fuse. So we can quite easily introduce different vesicles containing multiple targets of interest, which is markedly different than our traditional genome transplantation techniques. So through both of these chemical transplantation methods, I'm confident I have ample routes and optimization to install our genome without shearing it. And hopefully this will result in colonies forming on a plate and the world's first synthetic cells created from non-living parts. As a final thought, we do expect these cells to boot up. But what if it doesn't? One reason might be the chemical disequilibrium between the inside and the outside of the cell. So to alter that, we might need to start cells in a growth medium with different pH or concentrations of salts and nutrients. And instead of empirically trying all these different things, we can actually utilize Zanluthi-Schulten's computational models to better design a boot up medium. And we might need different transporters in the membrane, or we might need to add more. Like we can try with the E. coli secretory system, but if that doesn't work, the programmable fusion system makes it very easy to sequentially add more transporters. And we can adjust other parameters. When you think about combining a membrane and TXTL and a genome all together, it might take some adjusting. So you might need to lower the temperature and give it time or change the osmotic pressure. So those are some things we're thinking about, but I'm curious what else there might be and what are your thoughts? And with that, I would once again like to thank the Bessemer Trust and Ruggles family, as well as everyone else who's continuing to play a huge role in working towards this goal. It really takes a village to build and study a cell. Thanks so much, and here are my references.
Very nice, Tyler. Very nice. Questions?
Somebody raised their hand?
Or a question. Is it really nice? I mean, that's a super cool project, and if you get it to work, that'll be absolutely amazing. It has to work at some point. What do you think for kickstarting the cell? I mean, is it enough to just give it ATP to sort of energize everything, or are you going to have to actually like induce an electrochemical gradient, like sort of like one of these things you use to people have a heart attack? I mean, do you think there's something extra you have to do besides just?
I think not, because we've got really long-lasting protocells at this point, just with ATP. So I hope not, but you're right. That could be something.
Yeah. Cool. Thanks.
Yeah.
James, life, there's a sort of always a disequilibrium between inside a cell and outside a cell.
Right.
And that I think we certainly have to aspire to. But that's relatively easily done. Once you create a vesicle with a genome, you just move it into the right media, but that could take a lot of empiricism.
Exactly. Yeah. Trial and error. Right.
Absolutely.
Yeah. I have no doubt it'll work eventually, though.
Thank you.
I love your optimism.
Yeah.
Can you review all my proposals?
Absolutely.
We're going to learn quite a lot just attempting this. It's multifaceted.
That's also true. Yeah. It's fundamental insight.
Yeah. May I ask a question?
Yep.
Yeah, wonderful talk. I wonder about whether you could have control over the density of the cytoplasmic material. And I mean, as you know, even if you have good literature numbers on enzymes, their kinetics and activation energies change tremendously in what we call crowded environments. And the question is, do you have any control over crowding? So maybe your effect may work in one state, but not in the other, just by dilution or overcrowding whatsoever.
Yes. So in this cell-free system, Andre Sake does use Crowders, and he's kind of optimized the with PEG 8000 and the ratio of it to kind of optimize the protein yield. But yes, like once you contain it into the liposome, you might have to change it. You might have to use like a smaller PEG that can penetrate the liposome. There's definitely considerations for that. And the size of the liposome will affect the crowding too.
But is there any good experimental method established to determine this, except just adding PEG ratios, or can you control it after your reconstitutions and assay at least to know about the density, material density, or crowding effects in the final synthetic cell?
I think this is a very nascent field, and there might be some ways to concentrate it, but I think there's a lot of inconsistency with even for these cell-free expression systems. It's hard to make it consistent. So that's another thing. Like,
Yeah.
testing that might be hard.
Kareem,
Yeah, thanks.
this seems like an opportunity for empiricism. One of the advantages of the nitrogen cavitation is, as opposed to say, putting the cells into a hypoosmotic solution is and blowing them up, is here we can have minimal media or like no media and just work with cells and have a highly concentrated cell-free system without any dilution. That may work in our favor, or we might use osmotic methods to get rid of some of the liquid inside cells. They're tricks we can use. I see, in theory, this whole system is pretty straightforward, but in reality, there could be a lot of empiricism involved to make this work. Well, next year, I hope Tyler can be talking about, cells he's made.
It could
Okay.
be that quick. I think I heard somebody maybe have a question. Did somebody else have a question?
Oh, hello, it's me, Marco. Can you hear me?
Yes.
Hello. So, do you know if there's any studies or attempts to produce a pure system instead of TXTL cell structure-based system using only mycoplasma derived parts? Any kind of
No.
assessing
We're like,
if the 36 genes present in the classical pure are present in the mycoplasma, like the equivalents.
Andre Saka is the only one, to my knowledge, working on who worked on this, and he was trying to mimic this, make a mycoplasma pure system. Yeah.
Great. It's probably make your life easier if it would be available.
Right.